Good afternoon, everyone. My name is Babalo, based at the University of Cape Town, and I'm part of the H3aBioNet seminar series task force, or rather the webinar series, because we do these seminars online, so it's a webinar, and we run them monthly. So I'd cordially like to welcome you to the August 2019 seminar, and today's theme is going to be on symptom evolution following the emergence of maize virus. This talk will be given by Associate Professor Darren Martin, who is based at the University of Cape Town, the Computational Biology Department and IDM. This talk will be for about 40 to 50 minutes and will then allow at the end for Q&A session. So there'll be 10 to 15 minutes of discussion and Q&A at the end. However, if throughout the talk while Professor Darren Martin is speaking, you can type in the chat box. As you can see on your left, you can type your questions. And at the end, when you want to speak, so this is just some housekeeping. So instead of disturbing the speaker, either type in the chat box, and at the end, you'll be able to then ask your questions. There's an icon at the top with a human raising its hand or their hand, and you can click on that, and that will bring to us the attention that you need us to select you to ask your question. I'm just going to read a few lines from the abstract, because I mean it's a very comprehensive, and I believe that you saw the abstract in the advert, the merchant ad that we sent around a few days ago. So thank you so much for joining us. We do appreciate you taking time out to join us for this amazing talk, or rather what promises to be an interesting talk on symptom evolution following the emergence of maize virus. When a pathogen evolves, it is expected that natural selection will modulate the severity of any pathogen-induced disease traits that either directly or indirectly impact its transmission probability. Accordingly, for many pathogens, it has been demonstrated that the basic reproductive numbers are determined by evolutionary trade-offs between the pathogen-induced mortality rate and the transmission rate. I'm not going to read the whole abstract. I'll quickly now introduce Professor, or rather Associate Professor Darren Martin. Associate Professor Darren Martin completed his undergraduate studies at the University of Natal in Durban and obtained his PhD at the University of Cape Town, South Africa. He's currently an Associate Professor of Computational Biology in the Department of Integrative Biomedical Sciences at UCT. His research centers on the development of computational methods for extracting information from nucleotide sequence alignments, and is primarily focused on the detection and analysis of genetic recombination in viruses. He's also the lead developer of the computer program RDP4 that is widely used for analyzing genetic recombination, and has been actively pursuing both computational and wet lab-based research aimed at defining the adaptive value of recombination and the evolutionary constraints under which it operates. Without further ado, I would like us to all welcome and thank Professor Darren Martin for giving his time to join us. Thank you so much, Professor Darren Martin, for I'm sure it's going to be an interesting talk. Thank you so much for
thank you for the introduction, Pabalo. So what I'm going to be talking about today isn't really just related to maize streak virus, some obscure virus that infects a plant which, although important for feeding a large proportion of Africa's population, is not really something that is on most people's radar, unless you're a farmer somewhere out in the countryside. It's not really going to be that important to you. But what is important to almost everyone is the fact that viruses, by and large, are one of the main constraints on the survival of humans. They have been throughout human history, and they probably will continue to be major threats to human longevity into the future. All viruses, including maize streak virus, share a characteristic which is really important in their evolution. That at some point in the past, the virus had to have come into existence, and at some point after it came into existence, it's probably had to change hosts. Now, what that means is that viruses that start infecting one species don't always continue infecting that species. They will frequently switch to start infecting a different species from the one in which they initially evolved to infect. So what I'm going to be talking to you about today is following that process of when a virus switches hosts and starts infecting a new species, and the time thereafter using phylogenetics, this fantastic new research tool. I say new because it's only about 40 years old. Research tool that we now use to study the evolution of pathogens, all pathogens, from HIV through MSV, maize-streak virus. And what I'm going to do is I'm going to just give you a little bit of a background on, first of all, how my research fits into the phylogenetic community, the community of researchers around the world that are working on developing new techniques based on phylogenetic inference for extracting information from nucleotide sequence data. So what my research group does is it works on a range of different computational tools that are meant to enable and empower scientists throughout the world, but particularly scientists in parts of the world where perhaps wet lab research is a bit too costly, or the facilities aren't available to do it, where you can use data that's publicly available, free data, to do cutting-edge scientific research, just given access to the internet and a laptop or a desktop computer. So the computer programs that my group works on are all computer programs that can just run on standard desktop PCs, specifically PCs, not Macs or Linux computers, which most people don't have, but which can run on those other computers with suitable emulation software, which is also free. And all the software is free, of course. And the software that we devise leverages free, mostly analyzed data that's available in nucleotide sequence repositories such as GenBank. Now, GenBank has this enormous volume of data stored in it that it will take scientists hundreds of years probably to analyze if we stop gathering data today. The amount and the amount of data that's in GenBank is being dwarfed yearly by the amount of new data that's being added to GenBank. So there will always probably be this lag between the amount of data that's available and the amount of data that can be analyzed. And my objective, and that of many other people that work in this field, is to try and bridge that gap, or try and make that gap a bit smaller than it currently is, so that we can more effectively keep up with the amount of data that's being accumulated, not just for the sake of keeping up, but for the sake of getting the best value from that data with respect to saving lives and improving human conditions and improving just the well-being of life on this planet, basically. And I'm just going to spend a small amount of time, a really small amount of time, going into how that data is accessed, because I think some of you maybe aren't fully aware of how easy it is to obtain the data that you can use to produce, in a very real sense, groundbreaking scientific research at absolutely no cost. So the first portal of entry to that database is something that's actually familiar to virtually anyone that's done an undergraduate degree in science, is via something that you would know of as PubMed. Now, PubMed, most of you don't probably realize, or most of you have never probably bothered to explore the little bar on the top of the page of PubMed, where it says PubMed, and there's a little drop-down arrow next to that bar. And if you click on that arrow, you can change that arrow to nucleotide. And once you've changed it to nucleotide, every search that you type into the search box is now not going to return scientific papers, but it's going to return nucleotide sequence data. So if I typed in the HIV envelope sequences, I'd get a list of HIV envelope sequences, just like you'd get a list of scientific papers if it was on the PubMed field. And on the side of that bar, there's a rundown of what those sequences that are found are. And in this case, there are about 20,000 HIV envelope sequences have been found in GenBank. And on the side, it tells me that, well, about 16,000 of those are actually virus sequences. And because I'm interested in HIV, the virus, I'm not interested in things that look like HIV sequences that are integrated into the genomes of mammals or whatever, I will just click on that virus link, and it filters the results to only include virus sequences. Then, to save those sequences, I click on the Send To button on the top of the page, highlighted there, and I ask that the sequences be saved in FASTA format, for example. And that gives me a file, a sequence file, a flat text file that I can load into any of a number of different kinds of programs. But before I do that, I mean, 16,000 sequences is a lot of sequences to look at. So the standard size of phylogenetic style analyses that can be performed using the currently available tools is 16,000 is probably a little bit big for that. So what I'd probably like to do before I start trying to do any sort of phylogenetic analyses on those sequences is I would like to filter the sequences for a subset that is most appropriate for the types of analyses that I'm going to use. And what's most appropriate might be 100 sequences, 500 sequences. Sometimes 5,000 sequences might be an appropriate number of sequences to analyze. And there are these tools such as SDT, which is called Sequence Demarcation Tool, which have been devised to help you take that raw FASTA file and sort it into smaller chunks that can each be analyzed in a meaningful way. And then once you've got that chunk of data, there are other tools that can convert that raw FASTA file into a form that can now be analyzed using phylogenetic techniques. The first step in that process is to take those raw sequence files and to perform something called multiple sequence alignment. And to obtain a multiple sequence alignment is something that many tools can be used to achieve. The tool that we've devised at UCT is a tool called Impale. In my humble opinion, of course, I think it's the best multiple sequence alignment tool available. And it leverages other multiple sequence alignment tools. It doesn't just work entirely on its own. It uses alignment tools such as Muscle and MAFT to do the basic alignment. But where Impale really shines is that it helps a user create a data set that can be used to produce or to do phylogenetic analyses that are not going to be as affected by sequence alignment errors as if you don't use a tool like this. So the alignment part of the process involves lining up the A's, C's, G's, and T's of the nucleotide sequences so that all of the A's or T's or C's or G's that are in a single column of the alignment all have a single evolutionary origin. They're all homologous. They're all evolutionarily related. And in the alignment, part of the alignment process is adding gap characters to the nucleotide sequence files so that alignment of homologous sequences into columns can occur. And those gap characters represent insertions and deletions that have happened during the evolution of these sequences. Impale helps to reduce the number of false positives that can, or the amount of mistakes that can, or errors that can be produced in phylogenetic analyses by minimizing misalignment. And the way that it does that is it permits the user, because the computer programs that do these alignments are often not as accurate as we'd like them to be. Let's leave it at that. That usually a human being can improve those alignments given enough time and enough effort to fix the misalignments, the obvious misalignments that the alignment software or the different alignment programs make. And the way that Impale works and why it is so good is it gives a user constant feedback on whether the alignment is being improved or not, with a little graph at the top of the alignment program. So whenever you move nucleotides around and try to fix errors that would be made by the multiple sequence alignment program, it gives you feedback and tells you whether you've messed things up and made the alignment worse or whether you've made the alignment better. Anyway, once you have the alignment, that's where the real analysis starts. So everything up until this point was just basically producing a data set that's going to be useful to analyze in a constructive way with using these phylogenetics-based techniques. Now, another piece of software that we at UCT produce is something called recombination detection program that Babalo mentioned. And this is a computer program that can be used for various different things, but primarily for detecting genetic recombination. I'm not going to go into what genetic recombination is at the moment, but it's a type of evolution that viruses frequently use or frequently have. And it is important from a phylogenetic perspective because genetic recombination, if it's not properly accounted for, can undermine the accuracy of phylogenetic analysis. So, what you can do if you're interested in genetic recombination, though, and not just removing the effects of genetic recombination from data sets so that you can get accurate phylogenetic analyses, is you can actually analyze various different species and find evidence of genetic recombination, and that in and of itself can be publishable information. Many species are thought to not have genetic recombination, and if they do, if you can find some evidence that genetic recombination is a feature of their evolution, then that itself can be something worth publishing. But generally, what you want to do once you've got a data set and you've got a multiple sequence alignment that's reasonably recombination free is do other things besides just drawing family trees. And I'll get into a bit of detail later on what actual phylogenetic trees are, since I think maybe some of you aren't fully aware of what they are. But the phylogenetic trees in and of themselves aren't that important. What they are is a stepping stone towards more useful insights. So a computer program that people at UCT also work on, it's primarily developed at the University of San Diego, UCSD, in California, is a computer program called Hi-Fi for analyzing evidence of natural selection in nucleotide sequence data sets. And that evidence is, for example, used to identify sites in the HIV genome where the virus is evolving to evade drug resistance, or to achieve drug resistance, or to evade immune responses. These tools are used for working out where immune responses are targeting virus genomes. For example, where antibodies will bind to encoded proteins of viruses, or where T cells will detect little epitopes produced by the viruses that are detected by T cells. These things can be identified using signals of positive selection that are detected by Hi-Fi as a guide. And again, if you find something useful with these methods, it's often publishable in and of itself. No extra laboratory work is needed. Basically, just sequences downloaded off the internet and analyzed with these methods can be enough to produce worthwhile publications. Another method, we don't really work on it at UCT, specifically on coding the method, but we do a lot of testing of the method and application of the method, of the tool, is a computer program called Beast. And Beast is an interesting computer program because it uses phylogenetic analyses to do a whole range of things that other computer programs are incapable of doing. Among those are working out using phylogenetic trees to try and work out, for example, the population dynamics of species, usually viruses, but bacteria and potentially eukaryotes as well. Looking at movement dynamics of species, what their ancestral phenotypes were. It can infer based on the phenotypes of extant organisms what the phenotypes were of extinct ancestors of those organisms. And it does that by taking nucleotide sequence data together with a little bit of extra meta information, stuff like sampling times, the locations where those nucleotide sequences were sampled, and the phenotypes that those nucleotide sequences are associated with. And I'll be talking a lot more about Beast in the rest of the talk, but first of all, I'll say that these analysis pipelines can become quite complicated, and various different tools can be analyzed in sequence in these analyses that you do of these nucleotide sequences. You can, for example, analyze nucleotide sequences for evidence of secondary structures in the genomes of these viruses. The virus genomes frequently have single-stranded nucleic acid, frequently single-stranded nucleic acid molecules, and those single-stranded nucleic acid molecules can fold into shapes which are partially duplexes or partially double-stranded and have like helices like you'd expect in a normal double-stranded DNA molecule, but are partially single-stranded. And most commonly, you will have come across these sorts of things with, for example, tRNAs and things, the cloverleaf structures of tRNAs. You will have seen the secondary structures in that cloverleaf picture. And there's programs for inferring those secondary structures from just raw nucleotide sequence data. And there's programs for analyzing those secondary structures to see whether they're under selection or not using Hi-Fi. And there's programs for testing the results of Hi-Fi to work out whether there's sufficiently good evidence, or there's sufficiently good evidence to suggest that those nucleotides, those secondary structures, have some biological functionality. Things that are evolutionarily conserved are very rarely evolutionarily conserved for no reason. It's usually that they're evolutionarily conserved because they have some biological function. Anyway, so it's worthwhile asking, well, how can sequence data be so useful? How can sequence data contain so much information, more information than just what genes are encoded or how much of each gene is encoded? How can it encode so much information on the actual biology of the organisms? Like large-scale biology, like the phenotypes of the organisms and the population sizes of those organisms and things like that. And the reason for that is because living things evolve under selection. Things that work get maintained, and things that don't work get removed. And the frequencies of certain polymorphisms in a population, as well as the types of polymorphisms that occur, whether they occur in coding regions and are synonymous substitutions or non-synonymous substitutions, are all little breadcrumbs that let us figure out what the big biological processes are that have caused those signals, that are driving the types of nucleotide substitution, the types of genetic variation that we're seeing in the population that we're studying. And in many cases, those sorts of insights are actually only achievable using nucleotide sequence data. We can't analyze the evolution of organisms in real time, most organisms at least. I'll give you a big exception to that in a little bit because we actually do that with viruses. But for most organisms, we can't analyze their evolution in real time. And we have no other way of gaining insights besides maybe looking at fossils and things like that of how really at the molecular scale these organisms have become the way they are. Why are they the way they are? Why did they change from the way that they used to be? Why did ancestral forms of these organisms become extinct, and why were they replaced by the forms that we see today? And there's a huge story there as to how that sort of stuff happens. And that's what all this fiddling with nucleotide sequences and trying to extract all this information from nucleotide sequences, all that it's really about. So the idea of just only analyzing nucleotide sequences, free data, and getting publications out of free data, it begs the question, can you really just trust a scientific study that's only done on a computer? Surely you have to have some backup sort of laboratory tests to confirm what's happening. The inferences that you made are always just going to be inferences. They're not ever going to prove anything. But in reality, no scientific investigation ever really does prove anything. What we're doing is testing hypotheses and rejecting some and not rejecting others. And that's all these nucleotide sequence analyses are all about. Rejecting some hypotheses about the way things might have happened in the past and the way they might happen in the future, and finding evidence that's consistent with some hypotheses, not consistent with others. And if you do these sequence-based studies properly, the results are as reliable as any lab-based study that you could care to do. As long as, and there's a caveat here, as long as the data is good. So these studies can definitely replace wet lab studies. And like as I said before, there is no alternative in the wet lab to doing many of these studies. If the sampling of extant forms of the organism that you're studying, and by extant I mean things that have been sampled maybe during your lifetime, has been thorough enough, there's enough nucleotide sequence data available to analyze with these methods, and this data becomes more valuable with additional sampling time information, if it's known when these samples were taken, if it's known where in the world these samples were taken, and if something else is known about these organisms. What sorts of symptoms did they produce, if they're viruses or bacteria or pathogens? And other things about their phenotypes. Sort of, if you're talking about HIV, for example, what was the viral load of the person that this HIV genome was sampled from, or that HIV genome was sampled from? What was the T cell count of the individual from which that virus was sampled from? These bits of meta-information add tremendous value to the nucleotide sequence data. And tremendously expand what can be done with the nucleotide sequence data. And that's what I'm going to be focusing on with the remainder of the talk, is how phylogenetic trees is this really simple construct for explaining or describing the genetic relatedness of different individuals to one another, can be used to determine stuff like when evolutionary events happened in the past, for example, where you can time a phylogenetic tree, rather than it being scaled by genetic distance, it's now scaled by actual time, where ancestral nodes in that tree represent a particular time in the history of those organisms, a particular date or range of dates when that ancestral sequence existed. And if the metadata you have isn't just when the sequences were sampled, and sorry, I didn't mention that, you can do that based on knowing the dates when the sequences you're analyzing were sampled. You can infer into the past, if there's enough temporal signal in your data, your sequences have been sampled over a wide enough span of time. You can use that span of time over which those sequences have been sampled to calibrate a molecular clock, which then lets you work out that transformation from genetic distances to actual time when you're scaling the phylogenetic tree. And that's usually the first step, and that's why I put that first. But after you've done that, in addition to that, what you can do is, if the little bits of meta-information that you have that went along with your sequences were where those sequences had been sampled, for example, you can work out where the ancestral sequences of those sequences probably existed. So in this simple example over here, we have two sequences sampled from Mozambique. Sequence 2 and 3 are from Mozambique. And it's very plausible that the most common ancestor of those, most recent common ancestor of those two sequences was also sampled from Mozambique. But that little clade of Mozambique sequences is nested within a bigger clade of South African sequences. And it is very likely that at some point in the past, represented by the branch that links those Mozambique sequences to the rest of the tree, those sequences moved from South Africa to Mozambique. And we can use phylogenetic trees with a little bit of extra meta-information on where those sequences were sampled to work out where ancestral sequences existed in the past, geographically, where they occurred. And in very much the same way, we can work out not just where those sequences occurred and when those sequences occurred, but we can actually work out what the actual nucleotide sequences of those sequences were. We can figure out what those sequences were. We can infer ancestral sequences using these phylogenetic methods. And in very much the same way, if we know what the phenotypes are of the sequences that we've sampled today, at present time, or contemporary sequences, we can work out what the phenotypes were of ancestral sequences. We can work out how the viruses have changed in the symptoms they produce over time. And finally, and I won't be talking about this at all, we can also use these phylogenetic methods to infer with a lot of other meta-information based on where the sequences have been sampled, because there's usually information available on rainfall in those regions or temperatures in those regions, how connected the regions are by air or road or rail. And we can start testing hypotheses relating to how did the viruses move across that landscape? What were the motive forces for them moving across that landscape? What was the incentive for them to move from one region to another? And that's all achievable with properly annotated nucleotide sequence data. Nucleotide sequence data where we know where the sequences were sampled, we know when the sequences were sampled, we know what sorts of symptoms the sequences are associated with, and we can start doing all of this really interesting characterization of the biology of these organisms using the nucleotide sequence data and the additional metadata together. And the way I'm going to explain this or describe this, put into practice, is with a virus that we use as an evolutionary model. And I said before that it's not usually easy to see the evolution of organisms in real time. Well, in some cases it is, and with viruses it is, especially. Well, not all viruses, but certain types of viruses. And the virus that we use as our model organism is a single-stranded DNA virus. It's one of billions and billions and billions of different types of single-stranded DNA viruses that exist. They're probably the most common organisms on Earth. And they're very good at analyzing evolution under experimental conditions because these single-stranded DNA viruses have very small genomes. They're only about 2,700 nucleotides long, this one that I'm going to be speaking about. And they evolve very rapidly. And for example, maize-streak virus, the model that we use, is a recombination and mutation rates which are similar to pathogens like HIV and HCV. They evolve literally in real time. We can do experiments where, and I will show you an example of such experiments later, where we can recreate large portions of the evolution of these viruses in the laboratory. Even a year or two-year long evolution experiment with these viruses can yield extremely valuable insights into how the process of evolution occurs. But maize-streak virus is particularly useful as a model, even amongst other single-stranded DNA viruses, because it infects a complex multicellular host. Most of these single-stranded DNA viruses probably infect bacteria. There are a few that infect human beings, but they don't cause any notable diseases in humans. Some of them cause diseases in domesticated animals. Most of you that have dogs have probably had your dogs vaccinated for Parvo viruses. Parvo viruses are single-stranded DNA viruses. And almost all of us are infected with these single-stranded DNA viruses. They're called TT viruses. I think it's almost 100% prevalence in the human population of TT virus infections. And these viruses don't cause any notable diseases in humans. But this virus, maize-streak virus, does cause a notable disease in maize. It causes a disease with very distinctive symptoms in maize, and that's really important. And maize-streak virus, we also think of it as being interesting because we know that it emerged. It switched hosts, and by emerged, I mean it started off infecting one host, and then it changed to start infecting another host. The hosts that it started off infecting were probably wild, uncultivated grasses in Africa. And then when maize was introduced into Africa in the 1500s, somewhere between the 1500s and the late 1800s, maize-streak virus learned and adapted to infecting maize. So we know that it is an emergent pathogen. The first reports of maize-streak virus were in the very late 1800s, and it probably emerged probably sometime between 1870 and 1900 as a pathogen of maize, which is approximately at the same time that HIV emerged as a pathogen of humans. Anyway, the other important thing is that there are over 1,000 very high-quality maize-streak virus full genome sequences. And by very high quality, I mean these are far higher quality than full genome sequences that you get for organisms such as HIV. Each of these genomes has been sequenced from a cloned virus, from a single infected plant. They aren't PCR-generated sequences, where you're sequencing whole populations of viruses. These are viruses of individual virus genomes that actually have existed at some point in the past, and that's crucial. The other important feature of them is that they're so small that you can resynthesize these genomes from scratch chemically for only about $300, which is a big bonus. And it's very easy once you have a genome of these viruses in hand, cloned in a cloning vector, to introduce that genome into a plant and to get the virus reactivated and to cause an infection. It's very easy to work with in the laboratory. So what we do is we took all the MSV genomes, because I'm going to give you an example now of how this sort of thing worked, the results that I'm going to present in a bit. We downloaded all the sequences from GenBank, aligned them, did some recombination analysis, because as I said, these viruses have high recombination rates, accounted for recombination, and then we ran this through BEAST, giving BEAST the sampling times of the viruses and the associated symptom phenotypes for those viruses. So essentially, the first thing we were interested in is the when of the evolution of these viruses, the timing of when things happen during the evolution of these viruses. And what we can tell from those analyses is that the diversity of MSV, the natural MSV population represented by this phylogenetic tree over here, the root of the tree is represented by the dot along with that branch. I'm talking about the left tree at the moment. Represents about a 120-year span of evolution. And we can confirm that that is an accurate representation of the time span of the evolution of these viruses, because we ran evolution experiments on these viruses where we made clonal infections and then we monitored them over a number of years. One of those experiments lasted, or two of the experiments lasted 5 or 6 years, but one actually lasted 32 years. A 32-year experiment created a tree that's about as wide, about 1/4 as wide as the tree, the 120-year time span tree. That gives us a lot of confidence that the time span that we've inferred the evolution of these viruses has occurred over using BEAST is accurate, that the most recent common ancestor of those viruses is in fact approximately 1900. Around about the time, within 10 years of the first report of the virus, the most recent common ancestor of all the maize streak viruses that have been sampled over the past 30 years was probably a virus that existed in 1900. And we can be quite confident of that. Then we want to try and figure out, now we know the phenotypes of these viruses as well, and I'll explain to you in a second how we work those phenotypes out. But we want to try and figure out how those phenotypes have evolved over time. And the reason for doing that is that it's a big unanswered question with viruses, especially viruses that have emerged recently to start infecting a new host. What happens with the severity of those viruses? Theoretically, it's thought that those viruses should evolve to become less severe in the host that they've just started infecting. They may start off being very severe initially, causing a high mortality rate in the host that they infect, but eventually they should adapt to the host population and not kill their host, because it's not to the benefit of the virus to infect the host so severely that the host dies before the virus has any chance to be transmitted. So theoretically, it's expected that viruses are expected to become less severe. And that theoretical expectation is very widely, is a widely held belief amongst a lot of theoretical evolutionary biologists, particularly ones working with pathogens. But has never really been proven with in a real world situation, with real world viruses. There's a lot of circumstantial evidence suggesting that it's the way that it is, especially with some experimental evolution studies, but never has anyone actually looked at a real world emergence event, followed that over time, and observed this happening. And that's what we're going to try and do here. So maize streak virus causes these distinctive symptoms, and these symptoms are quantifiable. But more important than these symptoms being quantifiable, these symptoms are directly linked to the fitness of the virus, and that the virus can only be transmitted, acquired by its insect vector and transmitted from these symptoms. It has to be picked up in the symptoms and transferred to a new host from those symptoms. If the vector feeds on the green part of the leaf, it won't acquire any viruses for transmission. So the viruses colony that it produces in the plant is within those streaks that the virus makes on the plant. And if we can measure aspects of those streaks, and we can measure aspects of other features of the symptoms of the virus, we're probably going to be measuring something which is in some way related to how well that virus is adapted to survive in that host. So we do that by image analysis using a computer program called Diarrhea. And it's quite basic how this works. It's basic blue screen type image analysis. We take pictures of the leaves in a blue screen with a size standard so that we can keep this, we can measure not just the streak symptoms, but the degree of stunting of the leaves. And these are maize leaves that have been cut into a quarter. So we're only looking at one portion of a maize leaf. It's generally the second quarter of the leaf from the base of the leaf, so that the size of the leaf represents the degree of stunting of the plant, because maize plants are basically just all leaves. And by measuring leaf stunting, we're measuring whole plant stunting. And we can also calculate the distortion of leaves. Distortion is another symptom that we can measure. And we simply do that by looking at the deviation, because you can see these maize leaves are approximately a rectangular shape. And we look at basically just deviation from a rectangular shape, and use that as a measure of distortion. And we can also obviously measure the proportion of the leaf that's covered by chlorotic streaks, which is useful. We can measure the width of the streaks. We can measure the color intensity of the streaks. The whiteness of the streaks reflects the amount of chloroplast destruction that has occurred within the photosynthesizing cells that the virus infects. And all of that information is useful information with respect to trying to figure out how the virus has adapted to this host in the 100 years after its emergence in that host. And we did that. We measured the phenotypes in 60 cloned MSV isolates that were all infectious. And then we used that information together with the nucleotide sequence data in BEAST to try and work out how the symptoms had evolved over time. And that's what's reflected here in color. So you're seeing higher chlorotic areas are reflected by purple colors, and lower chlorotic areas are reflected by dark or olivey, muddy green colors. And as you can see in this phylogenetic tree, and it's also time scale, because like I said, we're using the range of sampling times of these viruses, which have been sampled over about 30 years, to scale the phylogeny not in genetic distances, but in time. So we can work out the timings of ancestral sequences as well. But over the past 100 years, very clearly you can see that BEAST has inferred that chlorotic areas produced by these viruses have increased. Very, very clear signal of that. But how accurate is that inference? Because that inference that the virus is getting more severe over time, which is the exact opposite of what theoretically should be happening. How accurate is that inference now? I mean, this is contradicting theory, right? So what we do is quite clever, actually. We use phylogenetics as well to infer what the ancestral sequences were of those viruses, which is something that is quite straightforward to do. And then we synthesize those sequences. We chemically synthesize them, and then we test those sequences to see, well, do the symptoms that they produce match with the symptoms that are inferred to be produced by BEAST? Is there some support that we can observe that the severity, or is there some support for the inference that the severity of these symptoms is getting worse as time goes by? And we can compare the observed symptoms of those viruses with the inferred symptoms of those viruses that were made by BEAST. And what we're expecting in these regression analyses, because this is just basic linear regression analysis, we're expecting, when we do a regression of the inferred on the y-axis and the observed on the x-axis, we're expecting the lines to all line up along the gray line running at 45 degrees. And when we do the regression line and we get the regression line, we're expecting that regression line to be approximately 45 degrees, passing through the x-axis and passing through the y-axis at 0. Okay, the y-intercept should be 0 if we've got perfect inference. If we've got perfect inference, we'd expect all of the dots to line up really on the grey line. But what we find is that the chlorotic area estimates, and I only showed the chlorotic area estimates in the previous slide, but all the other symptom measurements do have there's a positive correlation between the observed and the expected, but the chlorotic area and the stunting estimates seem to be really accurate, or reasonably, or sufficiently accurate for us to have confidence in the beast result. The observed symptoms of those viruses match quite well the inferred symptoms of those viruses with respect to leaf stunting and chlorotic areas. The other symptom types, the correlation isn't so great. The observations don't match the inferences so well. Okay? We're just going to focus really on the chlorotic area and the leaf stunting for now, where we seem to have quite a high degree of confidence in the beast results. And looking at those over time, we can see that, because we know when those ancestral sequences likely existed, all right, and we're calling these observed phenotype changes over time, because remember that these observed, these are observed phenotypes for viruses which have been inferred. It's a bit of a complicated concept, but we are not 100% sure that those viruses that we've inferred are the actual ancestral viruses. We're quite confident that they're very close to the ancestral viruses, but there are always 2 or 3 genome sites within any of this type of inference that you make that you can't be absolutely certain of. Nevertheless, all of these ancestral viruses that we resynthesized were infectious, and crucially, they confirmed the beast result that over time, the severity of these viruses with respect to chlorotic areas has increased. But when we look at the severity of these viruses relative to other measures of other symptom measures, we notice that there has actually been a decline in some of the symptom measures. So the chlorotic areas are going up, which confirms the beast result. The chlorotic intensity, the amount of which is a measure of the amount of chloroplast destruction that occurs within the streaks, has gone down. In that sense, the virus is becoming less severe. In terms of leaf deformation, the virus seems to be getting more severe. It's causing more deformation in recent times than it did when it first emerged as a pathogen of maize. And that is the one result that contradicts the beast inference results, which I didn't show you that result where the deformation data was overlaid on the phylogenetic tree like I did for the chlorotic areas, but that is a contradiction with the beast result. But the stunting measurement actually confirms the beast result, whereas for some of the maize hosts that we analyze the viruses in, we do see stunting decreasing over time, whereas in others we see it increasing. And just as an aside, we didn't bank all our results on a single maize genotype. We tested it on 3 genetically diverse maize genotypes, hopefully to recapture the diversity of maize genotypes that are grown throughout Africa or have been grown throughout Africa since the 1800s when maize streak virus first emerged. So what does that tell us? Okay, so the phylogenetic inference of symptoms has accurately identified, we didn't need to do all the lab work to do this, accurately identified that the amount of chlorotic area, the chlorotic areas that maize streak virus is producing, have increased over time since the emergence of maize streak virus. But they've also shown us that the intensity of the chlorosis in those streaks, the amount of chloroplast destruction that occurs within those streaks, has decreased over time. And what this is an indication of is something which evolutionary biologists absolutely love. It's the evidence of the existence of a trade-off, an evolutionary trade-off, in this case, between the number of cells in the host that the virus infects and the amount of harm that the virus is inflicting on the host. Because when the virus starts destroying chloroplasts within those streaks, it causes host stunting because less carbohydrates are produced from photosynthesis in those hosts. The host becomes less healthy, more prone to other infections, more prone to dying early. And if the virus is able to minimize the amount of chloroplast destruction that occurs within the streaks, so that even infected cells can continue producing carbohydrates, it seems as though the virus has maximized its probability of onwards transmission. So it's maximizing the degree to which it colonizes the host, while at the same time dialing back how much harm it does to the cells that it colonizes to ensure it maximizes its probability of onward transmission. And those trade-offs, although it seemed initially that what we were seeing contradicted theory about how viruses should be evolving, is in fact exactly how viruses are predicted to evolve when they start infecting a new host. There's something called the transmission virulence trade-off, which suggests that viruses should evolve to maximize, or to optimize rather, the amount of harm that they do to their hosts to maximize the probability that they will be transmitted. And that, for the first time, we have shown really with viruses that existed in nature. Whereas it's been shown previously with viruses that have been evolved in the laboratory. This, to our knowledge at least, is the first time that it's actually been shown in viruses evolving in nature. And obviously, this work involved a lot of different people, from the computer programmers that wrote the image analysis software, through the computer programmers that had to modify BEAST so that it would be able to do the phenotypic trait inference. And those people are all credited on this final slide, if anyone is interested in going through that and seeing who they are. Anyway, thank you very much. And if there's anyone that has any questions, I'll be happy to take them now.
I'm here. Thank you so much, Darren. I appreciate your time and effort giving this talk. We are open for questions and comments. If there are any pressing, I'll give actually 5 minutes or 10 minutes max, if there are any pressing questions. But otherwise, Darren is willing to respond to your emails if you have questions. But for now, can you please type your questions in the chat box or raise your hand? There's an icon on top, pointing at it now, I hope you can see, with a person raising their hand, and you can press on that and we'll select you. Otherwise, if you are uncomfortable with speaking, you can type. We'll give you some time to do that. Okay? And just to repeat, this talk is being recorded. It's going to be available on YouTube in the next few days. So you can share it with your networks as well, and rewatch it. I see Fatma is typing, so I'll give her some time.
So can I answer Fatima's question?
And then we'll have Emmanuel ask his later. Okay, thank you.
Okay, Fatima, yes, BEAST is used commonly with eukaryotic sequences, and you mentioned Beauty. Beauty is the computer program that is used in conjunction with BEAST, specifically to tell BEAST, which pieces of meta information go with which nucleotide sequence, which dates each nucleotide sequence was sampled at, what the phenotypes of those sequences were, the geographical locations where those sequences were sampled, and so forth. And it puts that information into a form that the computer program BEAST can easily understand. And yes, it definitely is used frequently with eukaryotic sequences. There's some very high-profile papers looking at, for example, the extinction of the Beringian bison. Looking at, for example, the extinction of the dodo and things like that. trying to figure out, the movement of brown bears between the Arctic and subarctic regions and things like that. So a lot of the phylogeographic and dating uses of beast, as well as the population demographic inference parts of beast, have been used for eukaryotes quite extensively.
Okay, thank you for that, Darren. She's still typing, so we'll check her question as well. But for now, I'm going to allow Emmanuel to ask his question. Emmanuel, go ahead.
Hello, can you hear me?
I can hear you.
Okay, from the phylogenetic analysis tree, you've showed that the severity of the virus has actually increased from the time it was first detected. I wanted to understand
Yes.
if this could be due to different mutations that the virus have gone through, or the graph actually showed only that there are probably different subtypes that have been difficult to manage and not necessarily the original virus that was detected.
Sure. Yes, definitely. So the reason that the tree has got branches that you can see is that there has obviously been evolution of the virus through time. And some of those, the evolution is, in the case of a tree, represents, or the branch lengths of the tree represent the numbers of polymorphisms that occur, or at least they do in a normal tree, not really in these time-scaled trees. But you can make normal, the first tree that I showed, where the tree looks like and is unrooted, it just looks like a crack. There's all those branches spreading out off a central point. Those branches are scaled according to genetic distance, meaning mutations that have accumulated along those branches to give the viruses that we see today. And that process, or some of those mutations, are, I'm sure, responsible for the changes in phenotype that we're seeing in the viruses. We're seeing changes, the increases in the amount of chlorotic area that the viruses are causing. Some of those mutations are responsible for those changes. We don't know which of the mutations are responsible for those changes, and it is actually something that I have students working on, trying to figure out at the moment. But it's surprisingly difficult, even if there have been a small number of mutations separating a virus that doesn't produce big chlorotic areas compared with a virus that does produce big chlorotic areas. Even if there's just a small number of mutations that differentiate those two viruses, it can be extraordinarily difficult to find out which mutation or combination of mutations are responsible for that phenotypic difference. But we've got some clever ways of trying to figure that out, and we are actually going to try and figure that out in the coming year or two. Thanks for the question, Emmanuel. It's a good question.
Thank you. Are there any more questions?
Anyone else
or comments? Emmanuel, do you have another question? Because I see you raise your hand.
Yeah. I wanted to understand, because from the diagram, you see that sub-Saharan Africa is actually the geographic area highly affected by the maize-streak virus. Is there environmental factor associated with that? Maybe they are able to survive much better in sub-Saharan Africa than in other parts of the world, or these other factors probably that is enabling the virus thickness within this geographical region. I'm not meaning the sounds like.
Yes. I didn't really have time to talk about that, but we've also looked at, because it's such a useful model, we've done everything with this virus. We've also looked at the factors that are associated with its movement around the continent. And the virus is only found in Africa, by the way. It's not found anywhere else in the world, because the insect that transmits the virus is restricted to Africa. It can't survive in Europe or Asia. But the grass species that the virus infects are found everywhere in the rest of the world. So the thing that restricts its movement of the virus to the rest of the world is the geographical range of its insect vector. But what we've looked at is how the virus moves around Africa, and what we can tell is that the places where the climate is good enough for there to be a maize growing season that lasts the entire year. So in the part of the map where South Africa, Zimbabwe, and Mozambique all join or share borders, is a region of Africa where you get what's called a 3-season year for maize production. Another part of the map where you get a 3-season year for maize production is the Great Lakes, around the Great Lakes in East Africa. And what we find is that every single subtype of maize streak virus that has emerged that has originated in the last 50 years has originated from those two regions and moved to the rest of the continent. So we get what are called cradles of maize streak virus diversity, which are at the confluence of Mozambique, South Africa, and Zimbabwe, and around the Great Lakes. And the rest of Africa is really what we call the grave of maize streak virus diversity. So the cradle is where the viruses are originating, and they're moving everywhere else. But there are waves of movement from those two places. So yes, we are quite well aware of the environmental factors that are associated with the origin of different maize streak virus strains. And we even know the rate at which the viruses are moving. They move at approximately 35 kilometers a year from those two cradle regions. And it's quite staggering, or quite striking how we can narrow down where these viruses emerge from, simply because they emerge so frequently from those same places over and over again and spread across the continent in these waves. Yes, that's another good question, Emmanuel. We've done that.
So for now, I'm gonna look, in the interest of time, I'm gonna ask that if anyone has more questions, that they email you directly, Darren. And just to remind them that the slides will be made available if they need them, and the fact that it's gonna be on YouTube in the next few days. I just wanna say thank you so much once more, Darren, for a very useful and interesting talk. I'm echoing what Fatma said, that for people that are working on phylogeographic data, this should be very useful. So we really want to say thank you so much for your time. And to everybody else, we're going to have another webinar in September, the last week of September, and we'll be communicating that in due time. But for now, join me in saying thank you to Professor Darren Martin for his time and effort. Thank you so much, Darren.
No worries, man. Thanks for inviting me.
Enjoy your day and see you next month. Adios, everybody.